Okay, can everyone hear me?
Yes.
Good, very nice. Thank you very much for the kind introduction. Yes, my name is Kees Weststrat. I will put a pointer here so I can show some things. And it's very nice to have this opportunity to talk at this wonderful workshop where I can present the work that we are doing, where we use surface science to study isotope synthesis on cobalt catalysts. And in my description, the chairman already introduced me, and he said I worked at TUE as the last, but then afterwards I moved to a new company which is called Syngaschem, where I work now for a couple of years already. Already yesterday you have seen a talk from my colleague who is performing the electrochemistry research in our small research company. We are located in the Netherlands. We're a small team of highly qualified researchers and some support people as well. And the research we do focuses on two topics. One of them is electrochemistry research. And about this, my colleague presented yesterday for Tiny Sapunzi. The research that I'm mainly involved in is the fundamental understanding of catalysis using surface science. So our small research company is actually part of a bigger research consortium. We work very closely together with Syncat at Beijing. This is the research laboratory of Synfuels China, which is located in Huairou, excuse my pronunciation, where Professor Hans Niemannsried and Yong Wang Li are the directors. And we are a small branch connected to their research. So the topic of my research mainly is the isotope synthesis, and I think many of you will be familiar with this catalytic reaction because it is quite important in industry, also in China. For example, Synfuels China uses this process really a lot. And this is the chemistry of isotopes. So you start with carbon monoxide and hydrogen, two simple molecules. They will react with a catalyst. I call this the black box because we want to know what's happening inside, but it's really complicated. And what comes out is the thing that you want to have. And you want to have long chains of hydrocarbons, long carbon chains, but you also have to make water because in the beginning you have carbon and oxygen in equal amounts. And in your end product you don't have any oxygen, so you have to remove the hydrogen. So this reaction has been used already quite a lot in the past, and also today it is used to make synthetic fuels because these products can be converted into synthetic fuels. And also in the future, we think this will be important because we still want to make these molecules, but we cannot use fossil sources. So instead we can do the same thing, only then we have to start with CO2 and hydrogen, then make syngas out of that, and then we can still do this reaction. So the catalyst used in this process, and my presentation will be about the cobalt catalyst, looks like this. It contains very small cobalt particles. And those cobalt particles, they are bound to a high surface area support, which is typically an oxide or a carbon material. And this material is there to keep those tiny particles apart, otherwise they will collapse and make one big block of cobalt. And also sometimes the support can help a little bit with the reaction you want. And if you zoom into a particle, you can see it looks like this. And you have to realize that for the catalysis, those molecules, the hydrogen and the CO and the reaction product, all of that happens on the surface of the particle. So the atoms inside the particle don't do anything, only the atoms on the outside of the particle are important. They do the catalysis. So this is why if we want to understand this, we really have to look at the surface of the material. But then if you zoom into such a particle, and this is particle models that people have shown in literature, you can see that the surface is heterogeneous, which means that there's different kinds of surfaces on such a particle. And here you can see a typical cobalt particle. You can see different surfaces are there. You will have flat surfaces that can have two different structures, and you can also have monoatomic steps on the surface of different structure. And the thing is that these surfaces, they are different, so they will react differently. Now, we want to understand what happens, and really to understand the details. It's too complicated if all of those surfaces are present at the same time in our system. We cannot really understand what's happening on the surface. So to simplify this, we can work with a single crystal. And here you can see a picture. A single crystal is a special form of a metal. Normally a metal will be polycrystalline. It will consist of many tiny crystallites. But with a special procedure, you can make a single crystal. Here you can see an example of a cobalt single crystal. We then cut it under a certain angle. Then you make a small slice of it and you polish it, make it very shiny. And then afterwards, here you can see an atomic resolution image of a copper surface. And it's a single crystalline surface, which means instead of having all these different surfaces, like you can see here, you select only one of them, and you have a very big surface which looks like this, and then you can study it. And then if you want to understand what happens on the particle, you can do, as I show here, maybe 4 or 5 different single crystals, and together you can understand what happens on the catalyst particle. So this is one thing. You want to have a very simple model system, a single crystal surface, and also you need vacuum for this. And the reason is this. Normally, we want to work with a clean cobalt surface in our studies. But if this material is in the air, it will be hit very many times by gas molecules in the atmosphere. And this is really very many, because in normal atmosphere, one surface atom is hit 10 to the power 8 times each second with a molecule from the gas phase. So if you have a clean metal surface, it will never stay clean very long in air, because of the gas molecules from the air, they will hit and they will bind to the surface. So we can solve this problem by reducing the pressure that we use to very low values, 10 minus 10 millibars. So then we have suddenly only 10 to the power minus 3 collisions for each surface atom. And this is long enough so we can work with a clean metal surface. And this is why we have to do our experiments in a vacuum vessel like this. And you can see there's many things sticking from the sides, and each of these things is a different analysis technique. And you need to have all of them connected to the same vacuum vessel, because you cannot simply, when your sample is clean, you need special in situ cleaning procedures for this. When it is clean, you cannot simply take it out and analyze it somewhere else, because you have to go to air and you will contaminate it. And then when you have this, you can use a number of analysis techniques that give you information about the surface of this beautiful single crystal sample that we've used. So you can use electron diffraction, for example, to learn about the structure of the surface and the structure of the molecules that sit on the surface. You can do desorption studies that tell you something about how strongly molecules bind to the surface. Infrared spectroscopy telling you about vibrations. Scanning tunneling microscopy to tell you about the structure. You can do surface analysis, elemental analysis with XPS that tells you which elements are present on the surface and which type of molecules are there, and of course theory methods to help understand what we're looking at. Now we work in vacuum, which is not only to keep the surface clean, but it also gives us very good control of what is on the surface. So we have a good vacuum, and we dose, for example, a little bit of CO inside the vacuum chamber, then we can put a small amount of CO on the surface, and we can control how much that is. Now, to understand what we see with our analysis tools, it's also very useful that we have a single crystal surface, because then it means that there's only one surface, so all the reactants on the surface will react in the same way, and we can understand very well what we're looking at with our spectroscopies. So this is the approach we use to understand the mechanism of isotope synthesis. And we want to study elementary reaction steps on a cobalt surface to know better what is happening and why cobalt is such a good isotope catalyst. So it's a really complicated reaction. You have to do many things to get from the reactants, CO and hydrogen, to the products, the hydrocarbons that we want. And we can look at each of them individually. So let's first look at what you can learn about the adsorption of carbon monoxide. We can use infrared spectroscopy to figure out where the CO molecule binds to the surface. And this depends on the amount of CO we have on the surface. So if you put a little bit of CO on the surface, infrared spectroscopy tells us that the CO molecules like to sit on top of the cobalt metal atoms of this close packed cobalt surface that we've been using here. But then if you increase the amount of CO molecules on the surface, they start to repel each other, and they will sit on different places. So here, in this case, they will sit on two different places. They will sit on top of metal atoms, some of them, and some others will sit in between three metal atoms in a threefold hollow site. Then there's yet another situation possible where the molecule can sit also on a bridge location, so in between two cobalt metal atoms. So it's actually quite complex how the CO molecules, where the CO molecule sits, and this depends on the coverage that we have on the surface. So in an experiment, we can see what happens when we change the coverage of CO. And of course, what you want to know is where do we expect the molecules to sit in real reaction conditions. So in the experiment, what we do is we are at a very low surface temperature, only 100 Kelvin. So there, all the CO molecules will bind to the surface and they will stay there because they cannot leave the surface. It's too cold for this. Then we can have a top view of many of such spectra that we record during heating. And what you can see here is at specific temperatures, there's a change. When we heat it, molecules will leave the surface. Each change means there's less molecules on the surface. And as a result, the CO molecules will occupy different sites on the surface. Now, this is in vacuum, which means that the molecules are there in the beginning, but they leave the surface and never come back when we heat it. But you can do the same experiment if in a high CO pressure, and high for us, 10 - 3 millibars is already quite high if you are used to work with this ultra high vacuum technique. What we can see exactly the same thing actually. You see the same transitions happening, but they happen at a higher temperature. This is because we now have an equilibrium coverage of CO. There's CO molecules leaving, but also new CO molecules coming, so you can have a higher surface coverage on the surface at a higher temperature. We can also use another technique that tells us more about the quantity of the CO molecules on the surface. And this we can determine by measuring the work function of our sample. And in this way, we can find out what the CO coverage is at a given temperature combined with a given pressure. So here you can see one experiment we did. We have a lot of CO on the surface at low temperature, but if we heat it in a very low CO pressure, we can see that the amount of CO decreases when the temperature gets higher. Now we can increase the pressure and do exactly the same experiment, and we can find more and more CO on the surface at a certain temperature. And this teaches us how it works. So we know, okay, from the trend, we increase the pressure, we go to higher coverage. We can also extrapolate this to real pressures and temperatures. That's the blue line here. This is what we would expect to have under real Fischer-Tropsch conditions. We can see that the amount of CO we expect to have on the surface is in this regime, about half a monolayer of CO on the surface. So the low vacuum experiments help us to predict what we can expect to have under the really high pressures that are used in the real catalysts. And it's important to know that there's actually quite a lot of CO on the surface during the reaction, because as we will later see, the hydrocarbons that we want to create in this reaction, they will react differently if there's a lot of CO on the surface. I will skip this to be a little bit faster. So we go back to our overview of all the reaction steps that take place during Fischer-Tropsch synthesis, and we've learned something about CO, which is that the CO coverage under reaction conditions would be half a monolayer, so one CO molecule for every 2 cobalt surface atoms. And under reaction conditions, the CO will sit on top of the cobalt surface atoms, but some of them will also sit in between 3 cobalt surface atoms. Now let's look at the next step, the adsorption of hydrogen. We also need hydrogen, of course, on the surface to react. Here the experiment we do is this. We are at a low temperature where we dose H2, so it will bind to the surface. The hydrogen molecule will break apart, and you create 2 hydrogen atoms on the surface. And then when we heat it, we look with the mass spectrometer and we determine the temperature where the hydrogen leaves the surface in vacuum. And we do this for different surface structures. You can see a couple of surface structures here. And what we can see is that the temperature where the hydrogen leaves the surface really depends a lot on the structure of the surface. And we can see when the temperature is high, it means this is where the hydrogen binds the strongest. We can see on the flat closed spec cobalt surface, the one that you have really a lot on the surface of a real catalyst particle, this is where the hydrogen binds the strongest. But on the other places that you also have on the surface, it will bind less strongly. So we can see that the hydrogen adsorption strength is different on different parts of the surface. But we can also learn more because we can learn something about the reaction to go from hydrogen molecule, a hydrogen-hydrogen bond, to two hydrogen atoms on the surface. What has to happen is that the hydrogen from the gas phase will come to the surface, and then it can either simply bounce off like a bouncing ball and leave the surface, then you will not create anything on the surface. Or the other thing is that it can break apart and that it can create two hydrogen atoms on the surface. And the ratio between those two processes, either bouncing off or dissociating, this is called the sticking probability. So the probability that the molecule that hits the surface will actually stay on the surface. We can measure this in an experiment where we use a different hydrogen dose. So we give a different amount of hydrogen first to the surface, and then afterwards we heat it. And in our desorption experiment, we can determine how much hydrogen came to the surface. So here you can see we increase the number of hydrogen, and then we can see more and more hydrogen stayed on the surface until you reach the saturation point, because at some point the surface is completely full. Now I can plot this here for the different surfaces that we study. We studied a very, very flat surface with this close packed, and we also studied the same surface, but now with some monoatomic steps on it. And a third surface also has the same terraces, but it has many, many monoatomic steps which have a kink. And we can see that the efficiency for hydrogen to dissociate on the surface is really increasing really a lot if we have steps on the surface. You can see for the same amount of hydrogen molecules dosed to the surface, on the stepped and on the kinked surface, we find many more hydrogen atoms afterwards. So this shows us that those step sites on a surface, which of course on a real catalyst particle you will have quite a number of such defects as step sites, they will really help a lot to adsorb the hydrogen. And this is the cartoon that we think is happening. You say on the flat terrace, it's quite difficult to dissociate hydrogen. Most molecules will simply bounce off and not react. But in the step site, it's very easy. So a lot of the hydrogen molecules will go to the step site, there they will dissociate. But we know that the hydrogen at the step site doesn't bind so strongly. So after dissociation, they will diffuse away to the terrace, and then this is open again. So next hydrogen molecule can come to the surface. And this is how we can fill up the whole surface quite efficiently when there's step sites available. So we've also learned a thing or two about the adsorption of hydrogen on the surface. We know that the defects on the surface help to dissociate the hydrogen. And we also find that hydrogen binds a little bit weaker on open surfaces compared to the closed packed surfaces. So there's really a difference in hydrogen adsorption depending on the surface sites. Let's now move to the next step. We have the CO on the surface, we have the hydrogen there. Now we want to break the carbon-hydrogen bond. So we want to dissociate the CO molecule. For this, we used XPS. This technique gives you information about which elements are present on the surface of your single crystal. And also, you can learn what type or the chemical state of this element. So here you see an experiment where we first start off with a clean cobalt surface. You can see this straight line, it's a background, and this is the region we chose, is the region where carbon is visible. So first we have an empty surface. We don't see any carbon. Then we put CO molecules on the surface at a low temperature and we heat it to around 350 Kelvin. So then afterwards, what we see is atomic carbon on the surface. So what has happened here is that CO molecules sit on the surface. When you heat it, most of them will leave the surface again, but some of them will break apart, put carbon and oxygen on the surface. And this is what we see afterwards with our XPS technique. Now you can see the size of this peak, but we would like to know how much is this. So then we compare this with simply putting a new amount of CO molecules on the surface. And these CO molecules on the surface give a big, big peak at this place. Quite a different place than where the atomic carbon atoms are. And from this, we know this is a third of a monolayer of CO molecules. So from this, we realize that the amount of CO that came from CO dissociation on the surface, amount of carbon from CO dissociation on the surface is really small. is only 1%. So really a small amount of carbon is only formed in this experiment. And then we look at the surface and we see, oh, we have 1% of carbon formed, but we also, on a real surface, we will have around 1% of defect sites, of step sites on the surface. So this tells us that, yes, we can break the CO molecule quite easily on the surface. You don't need a very high temperature, but you really need to have the step sites there. So this is the cartoon. CO will bind to the surface and it can easily dissociate into carbon and oxygen, but you need a step site for this. Good. So let's continue. We've now adsorbed CO, adsorbed hydrogen, dissociated carbon monoxide, created carbon and oxygen on the surface. So the next step is that we have to get rid of the oxygen on the surface. Oxygen is there in a large amount, and it's very important to remove it because if the oxygen stays on the surface, of course, the reaction cannot continue. So let's look at this process in more detail. So to remove the oxygen, we know it will react with hydrogen to form water, and this is a two-step process. The first step is to react to form OH species on the surface, and then the second step is that the OH species can react. Either they can react directly with the hydrogen, or instead you can have a mechanism where two OH species come together, a hydrogen is transferred so that you have one oxygen and one water molecule in the gas phase. So what we want to know is two things. First of all, how easy is it to remove oxygen from the surface? So if it happens to be a fast step, then it's no problem. It doesn't affect the rate of the Fischer-Tropsch reaction. But if it's difficult, it means that maybe the removing of oxygen from the surface is slowing down the Fischer-Tropsch reaction. So we want to know something about the rate of oxygen removal from the surface. And also, zooming in, we want to know from this two-step process, which of the steps is the most difficult one? So which is the rate determining step? And in order to do this, we again use our XPS technique. And now we look at the oxygen atoms that we have on the surface. So first, we put oxygen atoms on our cobalt surface. And we do this by dosing O2 on the surface. And we follow with XPS. And we see when we dose O2 on the surface, we see a single peak growing at this position. And when the peak is at this position, we know we have oxygen atoms on the surface, and it's not cobalt oxide. Cobalt oxide is actually different, and you can see this because it will appear at a different place in the spectrum. So we have atomic oxygen on the surface, and this is what we want to study. So we verify with XPS that we have the right form of oxygen. And then the next thing is we want to react this with hydrogen. But before we do this, we have to look at some reference spectra that will tell us in our XPS spectrum where the different species are. So here you can see at this place, you will have the oxygen atoms. They will appear at this position in the spectrum. But you can very well distinguish or identify if OH is formed, because OH species have an oxygen, but then the oxygen will appear at this place in the spectrum. Then you can also maybe have water, and water will be located in this position in the spectrum. And finally, we can also have carbon monoxide. Carbon monoxide also has an oxygen atom, and this will be located here. So these are the references, so we know what we see in our spectra, and then we do our experiment. We first put the black line 0.4 monolayers of atomic oxygen on the surface, and then we go to a special facility where we can do XPS at a really high pressure. Normally, XPS needs a low pressure to work, but at special facility at the synchrotron, we can work with quite high pressures. So we have this, and we put 10⁻³ millibars of hydrogen. Now we look and we see, okay, this is room temperature. We still have atomic oxygen on the surface. We don't have a lot of OH form. We do have a little bit of carbon monoxide, and this is there because it's a contamination of the hydrogen gas. So we didn't put carbon monoxide, but it can be there as a little contamination on the gas phase of hydrogen, and it comes to the surface also. Then we heat, and we look at a series of spectra in the top view. So you can quickly see where the species are and if their intensity changes. So what you can see here, changes in the spectrum. Important to notice here, if we heat to about 470 Kelvin, all the oxygen has left the surface. And we do this heating in the presence of hydrogen. So it means when oxygen leaves, it will have reacted with the hydrogen and it will have formed water, which goes away from the surface. Now we can zoom in, we can analyze this, and we can get the concentration of the different species at different temperatures. So what we see is this CO we had as a contaminant from the beginning. It simply goes away from the surface. It doesn't react with oxygen, it just goes away as carbon monoxide to the gas phase. Then we think we have a little bit of OH species, but really a tiny amount, and its amount doesn't change. So it doesn't seem to build up on the surface. The only thing we see is the atomic oxygen concentration that decreases because water is being formed. And from this, we don't see OH. It tells us about the mechanism. So we have two options. We can either have the OH formation being the rate determining, or the next step, the formation of water being rate determining. We conclude that this step must be rate determining. It's very difficult to make OH, but as soon as OH is formed, it will immediately react and leave the surface. And this is why we never see it on our surface, because when it is formed, it will immediately go away. So it will not build up in large amounts. Then we can increase the hydrogen pressure to higher than 10 minus 3 millibars, up to 1 millibar. And this is quite special for this type of experiment, to be able to do this measurement at high pressure. And this will teach us how easy or difficult it is to remove oxygen from the surface in the form of water. What you can see that we need around 400 Kelvin or higher to make water. So it means making water is not so easy on the surface. You need quite a high temperature to do this. So this is what we conclude about the reaction of oxygen with hydrogen. We see that the OH formation, the first step to attach a hydrogen atom to an oxygen on the surface, is the slowest step. And we also see that it's quite difficult to remove oxygen from the surface. And this means it may actually play a part as a rate determining step for Fischer-Tropsch synthesis. So that brings us to the last part and the most important part, because in the end, we do the whole reaction, we do the whole Fischer-Tropsch synthesis to make hydrocarbon chains. We have single carbon atoms in the beginning, and we want to grow them into a very long chain. We want to make very long chain hydrocarbons. And we have to do this efficiently. And efficiently means we want to make very long chains. We don't want to stop at 1 or 2 carbon atoms. We want to make them very long. So in order to do this, we have the species that make the chain, they have to stay on the surface quite long. Because if you have a chain there, maybe C2, it will sit there. It will have to wait for another C1 species to couple with it. So the longer this chain sits on the surface, the more chance it has to meet more monomers and to grow into a longer chain. So efficiency here means that actually species have to sit quite long on the surface. Only then can they make very long chains. So people in the past have thought about how does a chain grow on the surface? What happens? So in the past, they said, okay, we think we have a carbon atom. CO dissociates, we have a carbon atom. It reacts with hydrogens, and we have methyl species, CH3 species on the surface. Then we can also have CH2 species on the surface, and they can couple together to make an ethyl species on the surface. Then from here, you can have another CH2 attached to it to make a propyl species, and it can go on like this. And it can also stop growing, and this can be in two ways. They say one of them is if you lose one of the hydrogens, then you will make a propene product, an alkene, or you can add a hydrogen, then you will make a propane product. So this is what people said in the past, but as a surface chemist, we'd like to investigate, okay, this is all surface reactions. How do they work? Can we test this? Yes, we can do this. And the way we test this mechanism is to simply put those intermediates on our cobalt surface. It's again a close-packed cobalt surface. So we can put propyl species on the surface, and we can put methyl species on the surface. We do this at a very low temperature, and then we heat it, and we see what happens. So for the propyl, what we see when we heat is that at a very, very low temperature, this is 170 Kelvin. It's much, much colder than room temperature. Already at this temperature, the propyl species will react. It will lose one hydrogen, it will make the propene, and the propene will leave the surface. So at 170 Kelvin already, propyl will decompose to propene, which leaves the surface. So in Fischer-Tropsch terms, this is the termination reaction. The methyl species is a bit more stable. It will stay a little bit longer on the surface. Here we have methyl species together with hydrogen. What we see here at 260 Kelvin, bit higher temperature, but still below the room temperature, methyl will react with hydrogen and it will make methane. Now, what does this mean for Fischer-Tropsch synthesis? What we try to do is we take this temperature and we translate this to real Fischer-Tropsch. And real Fischer-Tropsch takes place at 500 Kelvin. So we take the rate constant, we extrapolate these temperatures, and we will find that a propyl species at 500 Kelvin will be so reactive that when it is there, it will, in nanoseconds, 10 to the minus 9 seconds, it will have reacted very, very, very fast. Also, the methyl species, it's a bit more stable, but still, it will take only a millisecond before the methyl species to react and leave the surface. But what does this mean? Well, this means that those type of intermediates, propyl, methyl, they're so reactive, they don't stay on the surface very long. And if they don't stay on the surface very long, they cannot do chain growth. Because to do chain growth, the species has to stay on the surface, it will have to meet a monomer, grow longer, then stay a little bit longer to have more and more monomers attached to it before it leaves the surface. So these type of intermediates that are part of the classical mechanism, they simply cannot work because they will leave the surface too quickly. So then what's our alternative? For our alternative, we want to study simply how hydrocarbons on the surface react. This is what you can study in very much detail with our surface science approach. And we have used C2HX species. So we start with ethylene, it decomposes, and what we want to learn is how it reacts, and we want to learn about stability of the species and how they react. And also, it's very important that we want to study how they react in the presence of carbon monoxide and hydrogen on the surface, because these species are also there during Fischer-Tropsch. And now for our mechanism, we said what we're looking for is we want to have surface intermediates that are very stable, surface intermediates that will stay very long on the surface, because that's what you need to make a long chain. Then it has time to grow longer. If it leaves too quick, it cannot grow longer. Also, of course, what you want is to make new carbon-carbon bonds. So the species we're looking for does not only have to stay long on the surface, it also has to be reactive to make carbon-carbon bonds and in this way grow longer chains. So this is the experiment we did. Again, we use XPS at the synchrotron. We look at the carbon region, and this gives us a lot of detail of how much carbon we have on the surface, but also which forms of carbon are present. So the experiment we start is acetylene, C2H2. It looks like this on the surface. And then we put also a little bit of carbon monoxide in the gas phase. We are at the low temperature, so carbon monoxide will also be present on the surface. And we also have hydrogen on the surface. So this is the starting point here. You can see at low temperature, this is what our spectrum looks like. And here you can see the cartoon. This is what we have. Now we start to heat. So when we heat, the temperature is higher, so more and more reactions will become possible. And indeed, we see that the acetylene starts to react. This we can know because the shape of the spectrum changes in this region. And from the shape of the spectrum in our XPS technique, we can see that now we have formed another species, which happens at around 270 Kelvin. The species that we form is ethylidyne, CCH3. So what happens in presence of CO is that this species is transformed into this species. That's one surface reaction that we see happening on our surface. Now the next step that happens at a little bit higher temperature is that this species, it is stable in a way because it stays on the surface, it doesn't go away, but it reacts. It reacts with another of these species, so two of these together react, and what they form is this product, 2-butyne. So actually what happens is they come together, they make a carbon-carbon bond. This carbon atom reacts with this carbon atom to make this species. So they actually make a carbon-carbon bond. They make a C4 species. So two very important reactions that we see happening on the surface. So here we are at a low vacuum conditions, but we also did the same experiment with a similar experiment with the same analysis technique, XPS, but now slightly different at higher pressures. So here we are at room temperature instead of the low temperature that we had as a starting point in the previous experiment. And we put first on the surface, again, the acetylene. And now we stay all the time at the same temperature, but we increase the pressure of hydrogen inside. Here in the beginning, we put some hydrogen there, so we have acetylene and there will be a lot of hydrogen around it, but nothing happens. Acetylene is unreactive, it's a very stable species. But now we increase the hydrogen pressure further, and I said before, hydrogen can have some CO contaminants. And we see that this is the case because in the XPS, we suddenly see carbon monoxide on the surface. But interestingly, as soon as carbon monoxide comes to the surface, the acetylene starts to react. And we know how it reacts because we have seen this before. We know that acetylene can make ethylidine, CH3 species. And an analysis of the spectra that we have, we can indeed confirm that yes, the first step is that ethylidine is formed. But then it doesn't stop because we are at a high enough temperature. So the ethylidine species that we have formed because of CO can react further. They can do this carbon-carbon bond formation that we've seen before. And at the end point here, we find the 2-butyne, this coupling product. So we see exactly the same reaction sequence as we saw before in our vacuum conditions. And here it's very clear and very important to realize that it's actually the CO that makes this happen. So CO influences the form of the carbon species we have on the surface. It makes ethylidine, and this ethylidine is the species that we want to have because the ethylidine is the species that can very easily make carbon-carbon bonds. Now, I have to emphasize here that yes, CO is there, but it's a promoter. So it's not CO reacting itself, it's just on the surface and it helps the other species to react. So it's not CO that helps, that reacts itself, it's really a promoter in this case. Then the same experiment with our high hydrogen pressures can teach us something else, because the endpoint of this experiment was this 2-butyne adsorbate on the surface, and we have hydrogen in the gas phase, and we are at room temperature. Now we started heating, and we were following the carbon signal while we were heating, and what we see happening is that the shape of the spectrum stays the same. So you see the same two peaks seen from the top. The only thing that happens at around 400 Kelvin is that the carbon goes away. Well, the carbon goes away. Actually, what's happening is that our product is leaving the surface. So what we see happening here is we have the starting point, a 2-butyne adsorbate. It adsorbs very, very strongly, so it cannot leave the surface. But we also have hydrogen. Now, this hydrogen at the high temperature can bind to the 2-butyne hydrogenation reaction. It will make a 2-butene, and the 2-butene, it doesn't bind so strongly. So as soon as the 2-butene is being made, it will leave the surface. So this is the reaction that we see. And why is this interesting? Well, it is, again, a reaction where carbon species on the surface leave, which for Fischer-Tropsch is a termination reaction. It could be a reaction that happens at the end when the chain growth stops. And it's good to see that this reaction is not easy. It needs 400 Kelvin. So it's a slow reaction, and it is good for Fischer-Tropsch because slow reaction to terminate is good. It means species stay long on the surface. You have a lot of time to grow long, and only after a very long time they will terminate because the termination reaction is very slow. So with all of this, we can now put together a mechanism, and you can see here a number of papers where we have on different occasions published about this and give different experiments, experimental proof for this. So this is what we propose, how chain growth happens during Fischer-Tropsch conditions. First, the most important C1 species for growth is methylidine, a CH species. In other experiments, we have seen that if you have a CH, it can very quickly react with another CH, and it will make acetylene. Well, acetylene, I've just presented a lot of things about acetylene. We know it's a very stable species. It will not easily leave the surface, but when you have hydrogen and when you have CO, acetylene will transform into ethylidine, the CCH3 species. Now, this one is quite reactive. Our experiment showed us that it can easily make a carbon-carbon bond. So in our model experiment, 2 of these species were reacting, but under Fischer-Tropsch conditions, you don't have so many C2 species, but you will have a lot of C1 species. So under Fischer-Tropsch, we think it will couple not with another ethylidine, but with a CH species. So when you do this, then you will make the propyne species. So you have C3. Then, like acetylene, we also have shown with experiments that this one will also, under the influence of CO, convert to propylidine. You will have again something like this, which can again couple with the CH, and it can grow further like this all the time, and then it flips from the alkyne to the alkylidine because of CO, CH can insert, and you can grow a longer chain. It's also good to realize that leaving the surface, which should be a slow reaction, we don't want to terminate so quickly, it is quite difficult. And this we also know from the experiment I showed in the previous slide. If we have a propyne adsorbate on the surface, we know it can hydrogenate to the propene, and this is how it leaves the surface, but it is a slow reaction. So this is good, because the intermediates in our chain, we know it can happen. We've seen them with experiments, and we also know that the intermediates, they will stay low on the surface because they terminate very slowly. So that puts together quite a long list of things that we have learned about the Fischer-Tropsch reaction by studying surface reactions. Well, you can see them again. We've learned something about CO adsorption, about the adsorption of hydrogen on the surface, about what type of site you need for dissociation of CO. We've learned about that it's quite difficult to remove oxygen, so this may slow down the reaction rate. And we've also looked at how chains grow on the surface, and we propose that it is alkylidine intermediates, the alkylidine mechanism, and not the classical alkyl mechanism that is responsible for chain growth. That brings me to the acknowledgements. First, I have to acknowledge many colleagues that I've worked with right now, but also in the past. My colleagues from Syngaschem, Hans Fredriksson, Fortuin Zapunzi, Professor Hans Niemannsverdriet, PhD students that work together with me at Syngaschem and at the Dutch Institute for Fundamental Energy Research, and Michael Gleason, also a colleague from there. Past co-workers who helped with the experiment, Ali Kizokaya, colleagues from CESL, my previous employer, Jonel Chobika, Jan van der Loosdrecht, Peter van Helden. The synchrotron facilities, these experiments really require a synchrotron to be performed, so we went to a number of places, different places in Europe to do this, and we were funded by the Dutch Science Organization in part. And last but not least, we really receive a lot of funding from Synfuse China to do our fundamental investigations in Fischer-Tropsch synthesis. And this is the last slide of my talk. I see that I have used my 45 minutes very well. So I want to thank you all for your attention, and I am very welcome to answer any questions.
Thank you for your nice talk. And we have quite a few questions online. And the first is that, is there competitive absorption of carbon oxide and hydrogen gas?
Yes, that's a very good question. Yes, we've investigated this also.
Absorption.
Yes. Yeah, I understand the question. A very good question. Yes, we've looked at this. Yes, it's quite complex, but they do influence each other. It's still an important question is how much hydrogen will be on the surface under reaction conditions, because indeed CO will bind, hydrogen will also bind, they will influence each other, but both can be on the surface together. We know this, but we're still doing more experiments to find out about this. But yes, there will be competitive absorption of hydrogen and CO, for sure.
Okay, thank you. The second question is can you use hydroxylated surface to check the RDS of the OH reactant with a softer oxygen?
Yes, again, an experiment. This is we did recently. So we were able to create OH groups on the surface and then to learn how OH groups react. So let me quickly go to one of the slides. You can still see my slide, correct?
Yes.
Yes, okay, good. I will show you this slide, this one. So here I talked about this. Actually, this experiment here, I can show this. This is a reference spectrum. And the way we make this is we put water on the surface. On the surface we use, it's a flat cobalt surface. Water does not dissociate normally, but we use a trick. We use electrons, and the electrons, they break apart the water. So in this way, we can still make a hydroxylated surface. And this is how we can actually make this OH species on the surface with a little trick to break apart the water molecule. And then indeed, we studied how these OH molecules react. And we see the OH species react. And indeed, we see that at a very low temperature, the OH will go away. It will break apart, or it will, two of them come together to make the water. So we know about this also. But this is possible. Yes, a very good question.
So next question is how to avoid the metal corrosions in the presence of syngas with high content of carbon oxide.
Metal corrosion, yes. I think that depends. So you can have, yes, it depends on which metal you have. So one thing is you can have carbonyl formation. Cobalt carbonyl formation is possible, but doesn't happen so often. Another thing is sometimes people think that cobalt oxidizes during the reaction, but we haven't studied this with our model systems, but from other work in the literature also on real Cobalt Catalyst, I think people see that oxidation is not really a problem for the Cobalt Catalyst. So that the Cobalt Catalyst doesn't seem to oxidize under reaction conditions unless you have really a lot of water. So it is possible with a lot of water, then you can oxidize the Cobalt Catalyst. But if you don't go to conditions where a lot of water is made, you can keep the cobalt metal active and also in a metal state.
Okay, the next question is how far is it from surface science to real catalysis?
Yes, that's another important question. I think what you can do, you can have very detailed experiments. I've shown you very detailed experiments, which are completely impossible on a real catalyst to get such detailed experiments. This is, of course, why we do the surface science, because this is how you can learn it. But then it's a bit similar. Maybe people have experience with density functional theory calculations. It's in a way similar to this. You make a very simple model system. You learn a lot of details that you cannot learn from the real catalyst. And then you have to think what it means for the real catalyst. And in my case, for example, I will show you another picture to explain better. So here, for example, I can study only a single surface in my experiment, but then I take the result and I think about it and I think, now I know on a real catalyst, you will have all of them together, but they will be in a certain amount. So you will have a terrace like this. It will always have a step side at the edge. So you will have, what does it mean? So you have to think afterwards, after you have your result, you indeed have to think afterwards in terms of the structure. So this is how a real catalyst particle looks like. So how can I translate my findings to a real catalyst. And the other one is, of course, if you go to higher pressure, is there something different? Can we extrapolate the results we find at the low pressure to the high pressure? Of course, some of the experiments I did are at higher pressure. So the ones towards the end, it needs the special equipment at the synchrotron, but this kind of experiments, they are at a higher pressure. So that's the pressure gap. You can, with instrumentation, you can overcome this, and you can also learn if pressure plays a big role. So far from my experiments, I still think that the thing I see at low temperature and low pressure, you can extrapolate. You can say, okay, this can also happen at the real catalyst surface. You have to really find your results and then think afterwards what it means for the real catalyst.
Okay, I think the next question is quite related to this one. It is there any case use surface science to guide the design of a real catalyst? I think you have talked a little bit in the previous questions.
Yes, that's another interesting question. Yeah. Yes, I think in zooming out a little bit, I think there's a few examples where surface science, such type of experiments, have helped to really propose something new for the catalyst. But in general, I think more often we know catalyst works, we know Fischer-Tropsch's catalyst work, of course, Cobalt can do this, and now we want to know more. We want to understand exactly how it happens. This is where surface science comes. But then I hope, but it's not so direct that you can say this, I can translate to this, but that by having the right picture in your mind of how the catalyst works, and then you design a catalyst, you keep this in mind. So you think a little bit about, oh, what can we do? You have new ideas of what to try for a real catalyst because you have new ideas of what happens on the surface of your catalyst. So this thinking, it's not always direct. Sometimes it can be direct, but sometimes it's more indirect, that you better understand really at a deep level how it works. And this can help you to think about what to do better with the catalyst.
Okay, thank you. Next question is step size. Step size is important for FT reactions. So yes. How to create more step size during catalysis preparations?
Yes. Okay. Let me. I can maybe make this bigger. So this is, of course, that could be indeed an example of, I say, yes, we see the step size are important. So how can we make them more of them? One thing that people look at is it is known that the size of the cobalt catalyst particles in preparation, you can make smaller particles or bigger particles. This is what you can do in a real catalyst to change this. And people see that this has an influence on Fischer-Tropsch. So this is maybe good or bad. If it's too small, it's not good anymore. So this can be done, and people have tried to understand this in terms of the number of step size. So we think that different size of the particle can change the number of steps. But maybe there I would like to have one more, just very quickly show this picture, because it's not, at least what we propose in our articles, it's not only the step size you need. So what we think is you need both of those things. So all of these reactions of the hydrocarbons, they take place on the terraces, on the flat part. So you need the flat part to grow the chain, but you need the step size to make the C1 species, to dissociate your CO. So you need to have both of them together. And this is why you have this island where you have the terraces where the chains grow, but the monomers, they come from the sides, from the edges of the terrace. And then the nice thing is you will have many edges on a single terrace. So all of these edges can at a very slow rate make your monomers, but all those monomers, they will go to the same terrace. So you will have very many monomer making sites that can work together, because all the monomers come together on this single terrace where the growth can happen. And only then can you be efficient with the growth, because it's also very important that Fischer-Tropsch is a really slow reaction. And you need many, many monomers to grow a long chain. So it can only work if you have places on the surface where you can have locally quite a lot of monomers coming together to make your long chain. So this is what we think. You not only need step size, you need the perfect combination of step size and terraces. And this is your optimal Fischer-Tropsch catalyst.
Okay. So the next question is why carbon oxide is not involved in the reaction? How carbon oxide promotes the reaction?
Yes, so of course carbon monoxide is there in the beginning. You want monomers, so you need to dissociate the carbon-oxygen bond, the CO molecule, to create your carbon. So this is the beginning. In my mechanism that I propose here, let me go to that slide. Yes, so in the mechanism I propose, carbon monoxide has to react somewhere here. And that's why you need the carbon monoxide. But the point I wanted to make is that it is there to make monomers, to make this species in the end, but it's also there, not really reacting directly in this case, but it sits here on the surface and it influences the way that the hydrocarbon growing chains react. So it has more than one role. It is there as a reactant to make the monomers, but it's also a promoter where it influences how the hydrocarbons react. And this is not so well known. Of course, everyone knows you need the CO, otherwise you don't have carbon to grow chains. But we don't think the CO molecule inserts. It's rather the CO dissociates first, and then C1 species, they couple. And we think this is the mechanism.
Okay, thank you. So although we still have questions, but the time is out, so we should stop here. So let's thank Dr. Kees John. Sorry if I pronounced your name in the wrong way. So
that's fine, no problem.
Okay.
Yeah. Okay, so thank you very much for having me. It was a pleasure to present.